When we think of the word genome, we often imagine a template, a fixed set of instructions that describes who we are. However, this metaphor isn't quite right. Think of it as less of a template and more of a dynamic system that not only stores our information, but also controls the way it's used and responds to signals from the cell and the environment. To understand why this is important, we have to first understand what the genome is. Basically, the genome is the complete set of DNA inside an organism. However, DNA on its own doesn't really do anything. A gene is only important when it's used, meaning when its information is copied into an RNA and then turned into a protein, which is the process that we call gene expression. Think about how all of the cells in your body have the same genome. However, your brain cells, your skin cells, and your muscle cells all function very differently. This is because all of your cells work with a very specific part of the genome while managing to keep the rest silent. Surprisingly, only 2% of the human genome directly codes for proteins. The remaining 98% was once considered to be junk DNA. However, now we know that this remaining DNA is no junk at all. A lot of the DNA that doesn't code helps to regulate. It works as a control system by turning genes on and off, adjusting how strongly they're going to be expressed, and deciding when they should be active, which goes to show that the genome is not exactly a template to be followed, but more of a control system. This more accurate understanding of the genome has helped how scientists approach it in medicine. The hopes for genetic medicine were very simple. They were to find the faulty gene, fix it, and then cure the disease. Many tools made it possible to edit DNA remarkably well, and they have already shown success in treating genetic diseases. However, many gene therapies have struggled. A good example of these is the therapies using embryonic stem cells. Embryonic stem cells are pluripotent cells, which means that they can be transformed into any other cell of your body. These cells will be modified in the lab and then transplanted to people with genetic diseases. However, this treatment wasn't successful due to the fact that your body and your immune system views them as foreign objects, therefore rejects them. This was one of the many problems while dealing with gene therapy. Other problems we face is that genes rarely act alone. One single gene can affect many processes in the human body, and changing it can cause many side effects. Another one is delivery. Getting the right treatment into the right cells at the right time in the right amount can be very difficult. All of these things have limited success, but because of them, genomic medicine is evolving. Instead of focusing on changing DNA structures, researchers are now focusing on understanding gene regulation, genome structure, and cell-specific behavior. New epigenetic treatments aim to reverse abnormal gene activity without changing the DNA at all. We have already learned how to read DNA, and now we're beginning to learn how to edit it, which shows that the genome is not only a template that shows our limits, but rather a living system that responds, changes, and adapts. And learning how to work with it might be one of the most meaningful scientific challenges of our time.